Number 15. The parasites in the latrine. In the 1980s, inside a limestone rock shelter in Piauí, Brazil, archaeologists were sifting through something most people would rather step around. It was coprolite, fossilized human waste, dried and mineralized, until it looked more like a lump of dark stone than anything a person once left behind. Researchers were cataloging pieces from a site called Boqueirão do Sítio da Pedra Furada, and one sample carried something nobody expected to find so far back in time. Under a microscope, thin oval shells turned up in the sediment. They were hookworm eggs, the same parasite still causing anemia and malnutrition across the tropics today. The coprolite that held them was radiocarbon dated to 7,230 years before present, give or take 80 years. That predates the Great Pyramid of Giza by nearly 2,700 years. Old feces holding old parasites is not, by itself, strange. Humans have carried worms for as long as humans have existed. The trouble is where these particular humans were supposed to have come from. In 1988, Adato Arroz of the Escola Nacional de Saúde Pública at Fundação Oswaldo Cruz in Rio de Janeiro published findings on hookworm eggs, recovered from other Brazilian coprolites, dated between roughly 4,900 and 1,300 years before present, working with Luiz Fernando Ferreira, and later with Carl Reinhardt of the University of Nebraska-Lincoln. Araujo pushed the question further than anyone had. If the standard account of how people reached South America was correct, this parasite should never have made the trip. That standard account is the Bering Land Bridge. The accepted Clovis first model has the earliest Americans crossing from Siberia into Alaska over frozen or near frozen ground during the last ice age. Hookworm larvae need moist soil at around 22 degrees Celsius, roughly the temperature of a heated room, to mature outside a host before they can infect anyone new. And Arctic crossing offers nothing close to that for any stretch of time worth mentioning. So Araujo and Reinhardt argued the parasite could not have survived Beringia, which leaves two options. Either early Americans arrived by a warmer route, coastal or trans-Pacific, or the accepted timeline for that crossing needs a second look. In 2001, a researcher named Fuller published a direct challenge in the journal Medical Anthropology, questioning the dating and the identification of the eggs themselves. Araujo and Reinhardt answered in the same issue, defending the work line by line. Neither side landed a knockout blow. Nobody is suggesting the parasites walked to Brazil on their own. Tempting as that image is, somebody carried them inside a living body, along a route archaeology still cannot fully name. That argument has never closed. No genetic study has settled which path those early South Americans took, or how a parasite that needs a warm climate ended up buried thousands of years deep in coprolite on a continent the textbook map says should only have been reached through the cold. The eggs are still there, mounted on slides in Rio de Janeiro, older than the story anyone has agreed on for how they got there at all. That argument stayed pinned on South America, and it wasn't finished. Much closer to the present, and further south along the same continent, another buried body was carrying a pathogen with an equally impossible passport. Number 14. The seal origin mystery. In the desert of southern Peru lies a mummy of the Chiribaya culture, wrapped and dried by sand that has kept her intact for close to 1,000 years. Her spine tells a story before anyone even opens the wrapping. A section of vertebrae has collapsed inward, fused where healthy bones should be straight. It is a signature injury, the kind doctors today call Pott's disease, and it comes from one thing: tuberculosis. For a long time, that diagnosis would have started an argument. Tuberculosis was supposed to be a European import, carried across the Atlantic on the ships that followed Columbus in 1492. A pre-Columbian case should not have existed. Then in 1994, researchers led by Wilmar Salo and Arthur Aufderheide extracted DNA straight from a lung lesion in a Peruvian mummy and matched it to Mycobacterium tuberculosis. The debate over whether the disease predated contact was over. The debate over where it came from was just starting. 20 years later, in 2014, Kirsten Boss and a team working out of the Max Planck Institute and Arizona State University sequenced 3 complete ancient TB genomes from skeletons in the Osmore River Valley on Peru's southern coast. What they found surprised them. These strains were not closest to any human tuberculosis lineage from the Old World. They were closest to Mycobacterium pinnipedii, the strain that circulates in seals and sea lions today. That made a kind of sense for a coastal population. Andean fishing communities hunted and butchered marine mammals for food and hide, and a spillover from seal to human, transmitted through handling infected meat, fit the archaeology cleanly. The coastal population living beside coastal seals now had a 1,000-year-old genome linking the two directly. Case closed, or so it seemed. Then in 2022, the same research group, joined by paleopathologists Felipe Cardenas Arroyo and Judith Arnett, sequenced 3 more genomes. One came from an inland Peruvian site called M6 Estuquina. 2 came from the highlands near Bogota, Colombia, hundreds of kilometers from the nearest patch of Pacific coastline. All three match the seal strain again. There are no seals near Bogota, and there never were. The researchers laid out three possibilities, and admitted none of them is confirmed. The bacterium may have jumped through some unidentified land animal acting as a go-between. It may have spread the ordinary way, person to person, riding llama caravans and trade routes up out of the coastal valleys into the mountains, or some combination of both happened, again and again, in patterns the genomes cannot yet untangle. The paper's own conclusion is blunt about it. More research is necessary. It's tempting to picture a single llama caravan as patient zero, hauling the disease over the mountains along with the trade goods, a coughing courier nobody suspected. The genomes don't offer that kind of story. They just confirm the strain made the trip, not who or what carried it. Tuberculosis still kills more people worldwide each year than almost any other infectious disease, which makes this less a museum curiosity and more a live case file with a 1,000-year head start, somewhere between a Peruvian shoreline and a Colombian mountain range. A seal pathogen learned to travel inland through hosts nobody has identified, along a route nobody recorded. The bone remembers the collapse in her spine. It is nothing to say about how the sickness found her. Sand kept one body's secrets in Peru. Number 13. Ötzi the Iceman's whipworm. Among the gear scattered around a 5,300-year-old body in the Alps was a scrap of leather thong, and knotted onto it were two dried, leathery knobs of fungus. Nobody packing for a hike brings along fungus for fun. The body itself had already answered the bigger question. On September 19, 1991, two German hikers spotted it melting out of the ice on the Similon Glacier at the Austria-Italy border, about 10,500 feet up, and assumed they'd found a lost climber. Radiocarbon dating said otherwise, placing his death sometime between around 3350 and 3105 BC. What the ice had kept went well beyond the body itself. Skin, tattoos, a full toolkit, a copper axe, a quiver of arrows, and inside him, Contenza's body had never finished digesting. In 1998, Luigi Capasso at the Institute of Human Paleobiology in Chieti, Italy, examined material from Ötzi's colon under a microscope and found eggs of Trichuris trichiura, the human whipworm, published in The Lancet. That made Ötzi the oldest confirmed case of the infection on record. A single female whipworm can lay up to 20,000 eggs a day, which is roughly the seeding count of a large arena, produced daily by one worm living inside one man. Heavy loads like that bring cramping and anemia, and whipworm wasn't the only thing riding along. In 2012, Albert Zink's team at the Euroac Institute for Mummies and the Iceman sequenced Ötzi's genome and found Borrelia burgdorferi in his blood, the bacterium behind Lyme disease, making him the oldest known case of that too. In 2016, Frank Meisner and Zink pulled a near-complete genome of Helicobacter pylori from his stomach, of a strain traced to Asia, had hardened arteries visible on CT scans, and one Copper Age man was carrying a parasite, a tick-borne infection, a stomach bacterium, and clogged blood vessels all at once. That's where the fungus comes back in. The two pieces threaded on his gear are Piptoporus betulinus, birch polypore, a bracket fungus, loaded with compounds that are both purgative and mildly antimicrobial. The tidy reading is that Ötzi knew he was sick and carried his own medicine cabinet against the worms in his gut. It's a good story, and it might even be true. But the fungus also works as tinder and as a dressing for wounds, and Ötzi left no note explaining which use he had in mind. There's a bigger gap underneath that one. Ötzi is essentially the only body of this age and this preservation quality that science has ever gotten to examine this closely. Every claim about Copper Age health, this is what disease looked like 5,000 years ago, rests on a sample size of one man's stomach. Was his pileup of infections ordinary for his time? Or was he simply unlucky? There is no second Ötzi to compare him against, and until the ice gives one up, there may never be. In Africa, another organism was pulling off something almost as strange, breaking through the same fatal barrier twice by two completely different tricks. Number 12. Sleeping sicknesses double origin. Drop a trypanosome into human blood and watch a clock start running. Within minutes, the parasite is dead, torn apart by particles in human serum called trypanosome lytic factors, built from a protein named APOL1. This is why cattle and wildlife across sub-Saharan Africa carry Trypanosoma brucei by the millions, spread by the bite of the Tsetse fly, while humans standing in the same swamp stay untouched. The parasite simply cannot survive inside a person, and that is the rule. Except twice, somehow, it broke the rule. Two subspecies of that same parasite learned to live in human blood anyway, and they did it in two completely different ways. Trypanosoma brucei rhodesiense, found across East and Southern Africa, evolved a surface protein called SRA that physically grips the business end of APOL1 like a straitjacket, so it can never punch its lethal pore. Trypanosoma brucei gambians, responsible for more than 97% of all reported human cases, took a slower, stranger path. It dulled the receptor that pulls in the killing particles in the first place, grew an enzyme to chew up whatever gets inside anyway, and stiffened its own membrane with a separate protein called TGSGP, so the pore, if it forms at all, cannot finish the job. Two parasites shared one enemy, yet they arrived at two unrelated solutions to the same lock. In 2016, a 17-author team led by Willie Weir and Anneli Cooper at the University of Glasgow sequenced 75 Gambian samples collected across West Africa between 1952 and 2004. Every one of them traced back to a single ancestor, one original parasite that made the jump into humans and then reproduced asexually ever since, and that much is settled. What the same authors could not settle is when, depending on which mutation rate they trusted, the jump happened somewhere between 750 years ago and 9,500 years ago, a window 12 times wider than its own low end, and wider than the entire span of written human history. Rhodesians turns out to be a stranger case still. Phylogenetic surveys have found it scattered across multiple, unrelated branches of ordinary animal trypanosomes, not descended from one common jump, but reinvented independently more than once, surfacing in different populations at different times for reasons nobody has pinned down. Left unchecked, this is not an academic footnote. The Uganda epidemic of 1900 to 1920 alone killed more than 250,000 people, close to wiping out a mid-sized city in 2 decades. Combined outbreaks across East and Central Africa in that era are estimated to have killed nearly 1 million. The official position is straightforward: convergent adaptation, likely tied to early agriculture and closer contact between humans and livestock, unfolding over a few thousand years. It is a reasonable story, but it just cannot say when the jump happened within a 12-fold margin of error. Cannot say whether the parasite's strange clonal lifestyle came before or after it learned to infect humans, and cannot explain why one subspecies needed to solve this problem again and again, while the other only had to solve it once. Case counts have fallen, but the parasite has not gone anywhere. It is still circulating, untreated, in the cattle and wildlife that share the same swamps it broke into before, waiting on whatever pushed it across the line the first time. Africa isn't the only continent where a single body broke a rule nobody thought could bend. On the shore of a Latvian lake, one man's teeth were about to make the same case, this time for the plague. Number 11. The lone Latvian plague victim. At a shell midden called Renčkalns on the Salaka River in northern Latvia, a young hunter-gatherer was buried alone sometime around 5,300 years ago. He was somewhere between 20 and 30 years old, and there was no mass grave around him. No sign that anyone else in his community died alongside him. He was dug up back in the late 1800s, packed into a museum collection, and largely forgotten until scientists returned to his teeth more than a century later. What they pulled out of his tooth pulp was DNA from Yersinia pestis, the bacterium behind the plague, and not some distant cousin of the Black Death pathogen either. It was the actual thing, just an extremely early version of it, one of the most ancient branches of the plague family tree anyone has ever sequenced. As far as anyone in Europe has confirmed, he is the oldest known plague victim on the continent, which means whatever killed him predates the completion of the Great Pyramid of Giza by roughly 500 to 700 years, and predates the medieval Black Death by something like 4,700 years, a gap longer than the entire distance from that pandemic to right now, multiplied nearly 7 times over. In 2021, Ida Andrade-Svaltuenia and her colleagues at the Max Planck Institute for Evolutionary Anthropology published his genome in Cell Reports. What they found was strange. His strain, catalogued as RV2039, was missing a 20,000-letter stretch of its plasmid, the section that carries a gene called YMT. That gene is what lets modern plague survive inside a flea's gut and clog its digestive tract, which is the entire mechanism behind flea bite transmission, the thing that let the Black Death spread as fast and as far as it did. Without it, this strain had almost no way to move the way its descendants later would. So how did a Stone Age hunter-gatherer end up infected with a disease that, by every genetic marker, could not yet ride a flea from host to host? That question got harder to answer in 2024, when Frederick Valur-Searsholm and Carl Gorans-Sjögren published a study in Nature tracing plague through 6 generations of a single Swedish farming family, buried in a passage grave called Frälsegården. 17% of the population they sampled carried the bacterium. 3 separate strains were circulating through that community within a 120-year window, reappearing across multiple generations of the same bloodline. Some prehistoric outbreak quietly stalking Stone Age Europe, jumping from hearth to hearth like an early draft of the Black Death rehearsing its lines, makes for a good story. Without a flea to ride, it likely never moved fast enough to earn the word epidemic, no matter how good that story sounds. The mainstream reading, in fact, is that these were rare spillover events, an occasional bite or a butchered animal, nothing that spread the way a real epidemic does. But repeated infection across 6 generations of one family does not look like an isolated accident. It looks like something that kept coming back. Nobody has identified how the bacteria move between people or between animal and person without the tool that later made it so lethal. Nobody can say for certain whether infections like these actually drove the population collapse of 30 to 60% that swept parts of Neolithic Europe around the same period, a die-off on the same scale as the Black Death itself, produced by a version of the disease that supposedly couldn't spread the way the Black Death did. Some researchers argue the wider data rules out a prehistoric pandemic entirely. Others argue plague was very much in the room. One man lies in ground that was already 500 to 700 years old by the time the last stone went up on the Great Pyramid, carrying an infection nobody has fully explained and no flea to blame it on. If a hunter-gatherer could catch a disease like that alone, with no outbreak around him and no witnesses left to bury, how many quieter infections just like his never left a genome behind at all? Most infections like his simply vanish, leaving nothing for anyone to find. Number 10. 28 unknown viruses in a Tibetan glacier. In 2015, a research team climbed to the summit of the Gulia ice cap, high on the Tibetan Plateau in western China. The elevation up there runs close to 22,000 feet, higher than any mountain in the continental United States, and only a few thousand feet shy of Everest's peak. They drilled down through the ice and pulled up long cylindrical cores, stacked layer on layer like the rings of a tree. Except these rings went back almost 15,000 years, which makes them older than farming and older than the end of the last ice age. The team boxed the cores and shipped them off the mountain before any of them knew what was actually frozen inside. Back in the lab at Ohio State University, a researcher named Zhuping Zhong took over. Before touching the ancient ice, she had to solve a much older problem in her field. How do you prove a signal in the ice is really ancient, and not just modern bacteria that hitched a ride on someone's glove? Zhong built a new decontamination protocol from scratch, shaving down each core in a clean room until only the untouched center remained. Then her team, working with senior researcher Lonnie Thompson and microbiologist Matthew Sullivan, sequenced whatever genetic material was left. What came back, published in the journal Microbiome in 2021, was 33 distinct groups of viruses. Only 4 matched anything already cataloged by science. The other 28 did not match a single known virus, family, or relative on record. So the ice had done exactly what an archive is supposed to do. It had held onto a snapshot of the microbial world from nearly 15,000 years ago, sealed at the top of the planet. A follow-up study in Nature, Geoscience in 2024, pushed the idea further, tracing how these frozen viral communities shifted alongside past swings between warm and cold climates, as if the glacier had been quietly keeping climate notes. Nobody asked it to keep. The sequencing runs out of road right about here, because having a genome is not the same as understanding an organism. Zhong's team could tell you the genetic code existed and could sort it into taxonomic groups by cold adaptation markers. What they could not tell you, for the overwhelming majority of these 28 groups, was what host they infected, how they survived a deep freeze lasting millennia, or whether anything resembling them is still circulating today. As one of the researchers put it plainly, science knows very little about what is actually living in these extreme environments. And that cap matters more by the year, because glaciers like Gulia are melting. Nobody currently knows how many more unknown viral groups sit frozen in ice sheets worldwide, or what happens to them once the ice that has contained them for 15,000 years finally lets go. The researchers were careful about it. They never called these viruses dangerous, and they never called them dormant threats waiting to wake up. They called it something quieter and honestly stranger, an open question about what the ice has been holding and what it will hand back once it can no longer hold it at all. Number 9. Malaria's disputed jump from gorillas. Picture a single drop of blood under a microscope. Red blood cells stained purple, and inside some of them a tiny ring, no bigger than a period on a page. That ring is Plasmodium falciparum, the deadliest of the malaria parasites, and in 2023 alone, it helped kill an estimated 597,000 people. Almost all of those deaths happened in sub-Saharan Africa. The parasite itself is old news to medicine. Where it actually came from was not. Falciparum belongs to a small family of parasites called Lavarania, and every other member of that family infects apes, not people. So in 2010, a team led by Wayman Liu and Beatrice Hahn at the University of Alabama at Birmingham, working with Paul Sharp at the University of Edinburgh, went looking for the source. They did not capture a single ape. Instead, they collected nearly 3,000 fecal samples from 57 field sites across Central Africa and pulled parasite DNA straight out of the droppings of 80 chimpanzees and 55 gorillas. What they found, published in Nature, settled one question completely. Every human strain of Falciparum found anywhere in the world today traces back to a single lineage carried by western lowland gorillas, not chimpanzees and not bonobos. It was one gorilla lineage. Jumping once, that became the common ancestor of every human infection that followed. On the family tree, human malaria doesn't sit next to the gorilla parasite. It sits inside it, a single twig on a much larger gorilla branch. The source is settled, but the date is not. And this is where 4 separate research teams, using 4 separate genetic clocks, cannot agree by a margin of roughly 100 to 1, the kind of spread that would end a career in almost any other branch of science. In 1998, a mitochondrial DNA study nicknamed Malaria's Eve put the jump at somewhere between 3,200 and 7,700 years ago. In 2002, a different team ran nuclear DNA instead of mitochondrial and got hundreds of thousands of years, not thousands. In 2011, Loy's team ran the numbers again and landed on a 95% confidence range stretching from 112,000 years to over 1 million years, with the midpoint sitting around 365,000. Then in 2016, a team including Hahn and Sharp again sequenced the whole genomes of Falciparum's closest ape relatives and found them roughly 10 times more genetically diverse than the human parasite. That kind of narrow diversity usually means a recent bottleneck, so this group argued for a jump within the last 10,000 to 50,000 years, timed to the rise of farming and denser human settlements in Africa, 2 of the most rigorous genome studies in the field. 5 years apart, disagree by an order of magnitude on the same single event. No fossil ever records a parasite, and no ancient text describes a mosquito bite, so the entire timeline has to be rebuilt from the genetic residue left in people alive today, which is exactly why every new sequencing method seems to move the date again. Somewhere between the last ice age and the deep past of archaic humans, one gorilla parasite crossed into one human body and started an infection that still kills close to 600,000 people a year. Nobody has found the year it happened, or the person it happened to. Malaria's crossing happened so long ago that the trail went cold before anyone thought to look for it. Number 8. The Ice Age zombie viruses. In a lab in Marseille, a dish of amoebae is dying, and nothing is touching them. No one has added a toxin or a drug. They are simply falling apart one by one under the glass. And the thing killing them was frozen solid 30,000 years before anyone thought to look for it. That thing is a virus, pulled out of permafrost from beneath the Kolyma River in Tukolka, Russia, from a sample drilled 30 meters down and taken in the year 2000. It sat in a freezer for over a decade before anyone thought it out. Jean-Michel Clavry and Chantal Abergel at Aix-Marseille University ran it against the culture of Acanthamoeba castellani, the standard lab amoeba, and it started killing on contact, exactly the way it would have done in the last ice age. They published it in 2014 in the journal PNAS and named it Pithovirus sibiricum. It is large enough to see under an ordinary light microscope, which is unusual because most viruses need an electron microscope just to become visible at all. A year later, the same sample gave up a second one, Mollivirus sibiricum, a round virus with a genome coding for more than 500 proteins. Then, in 2023, the same team went back to Siberia and came back with something older, Pandoravirus yedoma, recovered from permafrost 16 meters under a lake bed in Yakutia, dated to 48,500 years old. That is older than the cave paintings at Lascaux by roughly 30,000 years. Its genome carries around 2,500 genes. The flu virus carries about 10 to 15. In that same paper, Clavry's group reported 13 more viruses, pulled from 7 different permafrost samples, all still capable of killing modern amoeba cultures after tens of thousands of years in the ice. There's a part of this that should be reassuring, and it isn't quite. Every virus revived so far only infects amoeba. Researchers chose amoeba on purpose as a safe stand, and precisely so, this work could be done without risking a lab accident with something that infects people, which means nobody has actually tested whether a human or animal pathogen can survive the same deep freeze, because nobody has looked. Clavry has said it plainly. If an amoeba virus can survive that long, there's no clear reason a virus built to infect an animal couldn't do the same. Paul Hunter, at the University of East Anglia, takes the other side and says there's no evidence any such threat exists at all. Both of them are right, in a narrow sense, because the honest answer is that no one has checked. Permafrost is thawing faster now than it has in recorded history, exposing ground that has been sealed since before the last ice age ended. In 2016, a heatwave on the Yamal Peninsula thawed out a decades-old reindeer carcass and released anthrax spores that killed over 2,000 reindeer and infected dozens of people, including a 12-year-old boy who died. That pathogen was only decades old, not millennia. Nobody has an inventory of what else is down there. The ground is opening on its own schedule now, and whatever it has held since before there was farming or writing or anyone around to ask questions is simply going to surface. Quick one, since you've made it this far. If you're still with us this deep into the countdown, this channel was built for you. Hit subscribe. Now, back to it. Number 7. The Bronze Age Plague. That lone Latvian hunter-gatherer from earlier in this countdown turned out to have company. In 2015, Eski Willerslev's group at the University of Copenhagen screened over 100 Bronze Age skeletons scattered across Eurasia and found Yersinia pestis, the plague bacterium, in 7 of them, the oldest from Estonia, around 4,800 years old. None of those 7 carried the flea-adapted genes that let later plague ride from host to host either. So the question stopped being whether ancient people caught plague, because clearly they had. It became a question of how it spread, and when it turned into the killer everyone now recognizes. Then in 2018, Maria Spyru's team found something that flipped the timeline again. 2 genomes from a Russian burial, roughly 3,800 years old, carried the full flea-adapted toxin gene, which pushed the origin of bubonic transmission back over 1,000 years earlier than researchers had assumed just 3 years before. By 2022, a follow-up study found that same gene showing up independently in unrelated populations in Russia and Spain, at similar but not identical times, suggesting it may have evolved more than once. That is the trouble with this whole field, because every few years a fresh genome forces everyone to redraw the chart. Nobody has identified how the older flea-free strains actually moved between people, and nobody knows whether these were isolated infections or something closer to an epidemic. There was even a popular theory tying early plague to the wider collapse of Neolithic farming communities across Europe, until fresh genomic data pointed away from a pandemic without offering anything solid to replace it. The mainstream view holds that plague first spread by some quieter, forgotten route, then acquired its flea adaptation gradually, possibly more than once in different places, for reasons nobody can yet explain. But in 2025, researchers found a Bronze Age strain in a sheep, not a person, which means the reservoir for this disease, thousands of years before it earned its name, might never have been human at all. If plague's true home turns out to be some animal nobody has tested yet, the search for it stays grounded, at least within reach of a shovel. Number 6. The sealed lake under Antarctic ice. On the 5th of February, 2012, a Russian drilling team at Vostok Station finally broke through. Their bit had been boring downward for decades, coring through blue ice that recorded the planet's climate going back hundreds of thousands of years. That day, it broke through into something else entirely, liquid water sealed beneath 3,700 meters of ice, untouched by sunlight or open air, for somewhere between 15 and 25 million years. That is longer than the human genus has existed, by a factor of 6 to 10. The lake itself spans roughly 12,500 square kilometers, close to the size of Lake Ontario, though nobody has ever actually seen it, sitting as it does under an ice sheet about 4 times the height of the Burj Khalifa stacked on itself. To keep the borehole from collapsing under that pressure, the drilling team used around 60 tons of kerosene and freon as antifreeze, and that fluid would become the central problem. In January 2013, the team pulled up water that had refrozen onto the drill bit as lake water surged into the hole. Sergei Bulat, a researcher at the Petersburg Nuclear Physics Institute, analyzed the sample and found a bacterial phylotype his team designated W123-10. Its DNA only about 86% similar to anything in the international genetic databases. Bulat told reporters he was very sure it was an unclassified native microbe, adapted to a lake with no light, crushing pressure, and dissolved oxygen levels several times higher than a normal lake. Then came the complication, and it came from inside the same building. Vladimir Korolyov, who ran the genetics lab at that same institute, told Interfax the specimens belonged to contaminants, kerosene from the drilling fluid, human skin cells shed during handling, or residue picked up in the lab itself. 2 scientists at the same institute looked at the same sample and reached opposite conclusions on the record. The disagreement only widened from there. Scott Rogers at Bowling Green State University surveyed the accretion ice separately and logged over 3,500 unique gene sequences, roughly 94% of them bacterial, and the rest something else entirely. A later follow-up even turned up sequences closest to an Antarctic rock cod, in water assumed sealed off from surface life for millions of years. Either the seal has never been quite as perfect as it looks, or the contamination runs deeper than anyone can currently prove. By 2017, the Russian team conceded on the record that it was impossible with the data available to determine the lake's true chemical and biological makeup. A newer study using nanopore sequencing found the uppermost water layer looked microbial DNA free, which pulls the needle back toward contamination, though it still leaves the earlier finds unresolved rather than disproven. Officially, the cautious explanation wins by default. Everything strange in these samples is contamination until proven otherwise, and no sample has ever been proven otherwise. But that is a methodological standoff, not an answer, because every dataset so far, whether Bulat's, Rogers, or the nanopore study, comes from a different method, a different lab, and lands on a different conclusion, and neither better statistics nor the passing years have closed that gap. Nobody has a photograph of anything alive down there, and nobody has ruled out that something is, in a borehole 4 kilometers beneath ice that sunlight has never reached. Water that predates the entire human genus is still waiting for someone to find a way to ask it the right question without contaminating the answer. Number 5. The hepatitis B lineage that vanished. Right now, roughly 240 million people are living with chronic hepatitis B, which is close to the entire population of Indonesia, carried inside a virus most of them will never talk about. It kills about 1.1 million people a year, mostly through liver cirrhosis and cancer, putting it in the same rough range as malaria, even though malaria gets the headlines and hepatitis B mostly doesn't. You would think a virus this old and this common would have a settled history by now, and it does not. In 2021, a huge team led by Arthur Kotter at the Max Planck Institute for the Science of Human History in Jena screened the remains of over 130 ancient humans from Eurasia to South America. Buried in that data was the oldest confirmed case of hepatitis B ever recovered, a genome pulled from a body around 10,500 years old. What Kotter's team proved was that HBV has been riding along with human populations since the early Holocene, showing up in hunter-gatherers on 2 separate continents. The virus moved when people moved. When farming spread into Europe, the hunter-gatherer strains of HBV were pushed out by a new lineage that traveled in with the farmers, and that lineage ran the table across Western Eurasia for about 4,000 years before it declined and eventually disappeared. 3 years earlier, a team including researchers from Cambridge and Zurich, with Eske Willerslev among the senior authors, had already found the ghost in question. Mollmann's 2018 study in Nature recovered a dozen ancient HBV genomes from Bronze Age and medieval skeletons and identified a genotype that dominated Europe and Asia for millennia, but left no modern descendant. It left behind no weakened cousin and no rare regional holdout, only a branch of the family tree that simply stops. Here's the part virologists have not settled among themselves. Every method used to date HBV's true origin gives a different answer, and not by a little. Substitution rates measured from modern outbreaks run somewhere between 100 times faster and 10,000 times faster than the rates implied by the ancient genomes themselves. So depending on which clock a researcher trusts, HBV's actual first jump into humans could have happened 10,000 years ago, or hundreds of thousands, or arguably longer. It plays out like 2 surveyors measuring the same bridge and coming back with 10 miles and 10,000 miles, neither one backing down. Nobody has confirmed which animal HBV came from either. Related viruses turn up in chimpanzees, gorillas, orangutans, even capuchin monkeys, and the chimp and gorilla strains have been caught recombining with each other, which muddies any tidy single jump story further. So the official version holds up as far as it goes. Ancient DNA tracks HBV riding along with real documented human migrations for 10,000 years. And that part is solid ground. What it cannot do is name the founding jump, or explain why one dominant Bronze Age lineage, after outlasting empires, simply stopped existing while the virus itself did not. Number 4. Hyperdisease. In the grasslands of Wyoming, there is a sinkhole, barely wider than a doorway, that has been swallowing animals for tens of thousands of years. Fall in, and there's no climbing back out. One of the animals that fell was a bison antiquus, and it landed at the bottom around 17,000 years ago, where the cold, dry air of the cave kept it from rotting away to nothing. When researchers finally pulled that skeleton out of natural trap cave and tested it, they found something that should not survive 17,000 years in bone at all. It carried DNA from Mycobacterium tuberculosis, the same pathogen that causes tuberculosis today, along with the fatty biomarkers that prove active infection rather than contamination. It is among the oldest confirmed molecular tuberculosis evidence ever recovered from the fossil record, and it predates the invention of agriculture by more than 5,000 years. An infected bison could be a coincidence. So in 2006, Bruce Rothschild and Richard Laub went looking for a pattern, examining 113 American mastodon skeletons pulled from museum collections across North America. On 59 of them, 52%, they found the same grooved bone erosions tuberculosis leaves behind. Something was moving through Ice Age North America, and it was not being picky about hosts. A rate like that does not read as background illness. It reads as an outbreak, which is exactly what Rothschild and Laub argued. The infection was too widespread across too many animals to be merely endemic. It looked less like bad luck and more like the leading edge of something. Not everyone agreed the pattern could carry the weight being put on it. In 2004, Kathleen Lyons and her colleagues tested the closest real-world comparison they had. West Nile virus, a disease that does jump between species. They found it could bias mortality by body size in birds. What it could not do, they concluded in ecology letters, was wipe out entire genera the way the end of the Ice Age did. No known disease, ancient or modern, behaves the way this one would need to. And the scale here is not small. The die-off window researchers are talking about runs from roughly 50,000 years ago to 10,000 years ago, a stretch of time 2 to 10 times longer than all of recorded human history put together. 38 genera of large mammal vanished from North America in that window. The mammoths and saber-toothed cats now frozen mid-stride in places like the La Brea tar pits. In 1997, Ross McPhee at the American Museum of Natural History and Preston Marks at the Aaron Diamond AIDS Research Center gave this idea a name: hyperdisease, a pathogen carried harmlessly by one species that turns lethal the moment it jumps into another that has no defense against it. They laid out exactly what such a disease would need to do, and nobody has ever found it. No pathogen isolated, sequenced, or confirmed to have existed, only the damage it may have left behind in bone. Most researchers today lean toward a combined explanation: Clovis-era hunting pressure stacked on top of the climate collapse of the Younger Dryas. That combination still struggles with details, though, like why megafauna survived dozens of earlier ice ages but not this one, and why the extinctions hit different continents in different centuries rather than all at once. Disease keeps getting invoked exactly where those explanations run dry. The lesions are real, and so is the DNA in that bison. But what actually carried it from animal to animal across a continent has never been held in anyone's hands, and there is a real chance it left nothing behind to find. Hyperdisease, if it existed, left nothing behind for anyone to hold. Number 3. The 250-million-year-old bacterium in salt. Half a mile under the New Mexico desert, near the Waste Isolation Pilot Plant, there is a layer of rock called the Salado Formation. It is salt, pink and translucent, laid down when an ancient sea evaporated. Inside some of those crystals are tiny pockets of trapped liquid, sealed the moment the salt formed around them. Researchers drilled a core sample from that layer and carried it back to a lab. One crystal, no bigger than a fingernail, held a droplet of brine that had not touched open air since before the first dinosaurs walked. They cracked it open under sterile conditions, drew out the liquid with a needle, and found a bacterium inside it. It was alive, and under lab conditions, it grew. The salt around it was 250 million years old. Russell Vreeland, William Rosenzweig, and Dennis Powers published the find in Nature in October of 2000. They named the strain 293, a halotolerant bacillus that had apparently sat dormant inside its crystal since the Permian period, waiting for a needle to let it out. If the dating held, this organism had gone into stasis roughly 5 million years before the first dinosaurs existed and woken up in a modern lab, cultured in nutrient broth like any ordinary sample. The claim did not sit quietly for long. In 2001, Dan Grauer and Tal Pupko, publishing in Molecular Biology and Evolution, ran the organism's genetic sequence against its closest modern relatives. The two were nearly identical. That is the part that bothered them, because an organism isolated from the rest of life for 250 million years should carry an enormous backlog of random mutation, the genetic equivalent of 250 million years of unanswered mail. Powers and his colleagues pushed back in turn, and by 2005 the argument was still running in the pages of geology, neither side conceding. Underneath the disagreement sits a harder problem. Unprotected DNA breaks down into fragments under 100 base pairs within about 10,000 years in warm conditions, and even in cold, dry conditions. The ceiling is closer to 100,000. 250 million is not a stretch past that limit. It is a different order of universe entirely, and there is no way to independently date the organism itself. Only the rock it came out of, the salt, can be dated. The bacterium cannot. So the debate has nowhere to go, because neither side has a tool that settles it. The same question already came up earlier in this countdown, in permafrost, far colder and far more urgent than a salt mine, where researchers have thawed out a virus that still infects a living cell after tens of thousands of years frozen, and watched a decades-old anthrax outbreak take a life the moment the ground gave it back. Cells can wait a long time in the cold. Nobody agrees on how long, or on what else is still waiting. Whatever else that salt crystal proves, it settles nothing about how long a microbe can lie and wait undetected. Number 2. The missing origin of syphilis. In 1495, French troops laying siege to Naples began falling apart in a way nobody in Europe had ever recorded. Physicians described pustules spreading over the whole body, flesh dropping from the face in chunks, and men dead within months of the first sore. Within a year, the disease had reached Holland and Greece. Within 2 years, it was in England and Scotland. By the turn of the century, it was in Hungary and Russia. Nothing in Europe had ever moved that fast. That disease was syphilis, or at least the ferocious first contact version of it, because the syphilis doctors treat today is a slower, gentler descendant of whatever tore through Naples that summer. And the question that has occupied historians and geneticists ever since is simple to ask and brutally hard to answer. Where did it come from, and why did it suddenly turn so vicious? For most of the 20th century, the answer looked settled. Columbus's crew sailed home from the Americas in the early 1490s, and syphilis appeared in Europe a few years later. The case looked closed, more or less, and historians loved nothing more than a tidy villain to blame. Then the ancient genome started talking back. In January 2026, researchers reconstructed a treponeme genome from human remains. The bacteria was 5,500 years old, and it sat at the very root of the family tree before the split that eventually produced syphilis, yaws, and Bajal. It proved the pathogen family is ancient and American, deep into prehistory, long before anyone crossed an ocean. 2 years earlier, Kurtu Meyander and Verena Schunemann, working out of the universities of Basel and Zurich, had already pulled 4 genomes from pre-Columbian Brazilian skeletons, one of them a tibia fragment with a telltale pitted bone of chronic infection. Those genomes match Bajal, the disease's non-venereal cousin, not syphilis itself. Together, the 2 studies build a strong case that treponeumal disease has been living in the Americas for thousands of years. But here is the part that never gets fixed. Every genome recovered so far that predates 1492 on either continent belongs to the mild, non-venereal branch of the family. Not one belongs to the sexually transmitted epidemic strain that leveled Naples. Kristen Harper went back through Europe's supposed pre-Columbian syphilis skeletons in 2011 and found the evidence for nearly all of them too weak to trust. So neither continent has produced the one thing that would actually end the argument, an ancient genome of true venereal syphilis, sitting there before the siege of Naples, waiting to be dug up until someone finds it. The story keeps splitting the same way it has for a century. Mainstream researchers lean toward an American origin for the wider disease family, with a European mutation into virulence after contact. A smaller camp points to a 2020 genomic study showing surprising strain diversity in early European skeletons, hinting the pathogen might already have had a foothold before anyone set sail. The disease that emerged from that argument still infects an estimated 8 million adults a year. Somewhere, in a graveyard nobody has excavated yet, the genome that would settle the whole thing is either waiting or it never existed at all. Losing one genome to time is a small kind of erasure. Something far larger vanished around 40,000 years ago. An entire branch of humanity and disease sits at the top of the suspect list. Nobody can convict or acquit. Number 1. The disease that may have erased an entire human species. If your ancestors came from outside Africa, somewhere between 1 and 4% of your genome is not, strictly speaking, human but Neanderthal, and it has been sitting in your DNA this whole time. A small inherited fragment of a species that supposedly died out around 40,000 years ago. That number is the whole mystery in miniature. Something wiped out an entire branch of humanity, and yet a piece of it is still walking around today. Reading this sentence through your eyes. Neanderthals were not a footnote species. They held Eurasia for over 300,000 years, roughly 10 times longer than the span of recorded civilization. They survived ice ages, hunted mammoths, buried their dead. Then, in a window as short as 15,000 years, they vanished. The last confirmed refuge sits at Gorham's Cave on the coast of Gibraltar, a limestone mouth opening onto the sea, where the final small groups appear to have held on before the record goes silent. In 2010, Svante Pääbo's group at the Max Planck Institute for Evolutionary Anthropology in Leipzig sequenced the full Neanderthal genome. The work later won Pääbo the Nobel Prize. It proves something nobody could ignore, that Neanderthals and modern humans interbred, which meant the 2 species did not just cross paths. They came close enough, often enough to exchange genes and everything else that travels alongside genes. That detail is what pulled researchers toward disease. In 2016, Charlotte Holdcroft and Simon Underdown proposed that incoming Homo sapiens carried pathogens out of Africa that modern humans had spent generations evolving resistance to. But Neanderthals, isolated and genetically narrow, had never met. It was less an invasion than an immune system arriving with credentials the other side had never seen and had no way to check at the door. Herpes, tuberculosis, and forms of stomach bacteria could travel along quietly, because a disease that one population has lived with for generations tends to stay mild, while the same disease dropped into a population, meeting it for the first time can turn catastrophic. A separate 2016 study out of the Max Planck Institute found Neanderthal-derived immune genes still present in modern humans today, evidence that Neanderthals carried their own adaptations to a Eurasian microbial world entirely different from the one arriving with the newcomers. Here is the problem nobody has solved. No ancient pathogen DNA has ever been recovered from a Neanderthal skeleton. Not one confirmed case, anywhere, in 40,000 years of remains. The entire disease hypothesis is built on genetics and inference, on the historical pattern of what happens when an isolated population meets a new one carrying diseases it has never faced. It is argument by analogy, not a body with the evidence still in it. The competing camps have not closed the gap either. Researchers arguing for climate change and resource competition published modeling in 2020 showing Homo sapiens simply outcompeted Neanderthals for scarce food. Then, in 2026, newer analysis found that climate shifts alone cannot account for the extinction timeline, and neither can competition alone. Every explanation runs out of road before it reaches the finish line, so the field is left holding several partial answers and no complete one. Disease remains one of the strongest candidates precisely because it is the hardest to disprove. it leaves almost nothing behind to find. Somewhere in that gap between one species ending and another carrying pieces of it forward, the actual cause of the disappearance is still out there, uncaught, unnamed, and possibly unfindable.